Great, thank you for that introduction. So to start off, thank you. I am a student in the statistics department at the University of Washington. I'm a statistician who works on statistical methods and tools for the analysis of microbiome data, which is why I'm here talking to all of you. And today I'm going to present a visualization tool to help understand microbial phylogenies, comparing and exploring them. So to start, I want to think about two different scales at which we might study microbial evolution. The first is at the genome scale, and this is what I see as the most popular way to study microbial evolution because it's the scale at which organisms can be compared. So we could say this is how microbe A is related to microbe B, and they are more closely related to each other than microbe C. But also this is a summary of how elements within a genome are evolving, because evolution is not acting on a genome as a whole, and there could be different evolutionary pressures acting on different parts of the genome. So if we zoom in to one of these parts of the genome, we could also think about evolution at a gene scale, the scale at which traits can change, as genes provide instructions for the construction of proteins. And we know that genes within a genome may evolve differently from each other. So I want to think about a way to compare these two different scales. And here, in these pictures, I have data which we might use to study this evolution. So here, for every organism we want to study, we could collect genetic information, and then often at the gene level, genes will be identified and then aligned together into a multiple sequence alignment. So this can be used to study evolution for a single gene, but often for a genome, these gene level alignments will be combined together through concatenation, essentially just pasting them next to each other. And then this can be used as a proxy for a genome. So once this data is available, often it's used to estimate phylogenies because phylogenies are the most common output used to understand microbial evolution. And as a reminder about phylogenies or phylogenetic trees, these are graph structures that are used to as a model for evolution. And here on the outside of the tree, we have tips. These represent organisms that we were able to sample and get these sequences from. And internal to the tree are nodes that represent ancestors. And the structure of this tree explains how different tips are related to each other. We also, in these trees, have the edges are called branches, and the lengths of them, if we have weighted trees, tell us how much evolution or how much genetic differences there are between two different tips. So this is a lot of information that's all encoded into this tree. And we can estimate our phylogeny at the genome level, or we could do it at the gene level. And as we often have multiple sequence alignments for many genes, because these are combined to create a concatenated alignment to come up with a genome level tree, we could instead estimate multiple gene level trees. And what we have here is a lot of data. We have all of these different trees made up of branching structures and branch lengths, and it's difficult now to compare them to each other. If we had just 2 or 3 trees and want to compare them, we could do this visually or through summary statistics. But as we have more and more genes, so we have more and more trees we want to compare, and as the number of tips gets larger, this is just going to become very computationally large. So this motivates this work, which is a visualization method to compare gene-level phylogenetic trees, which we'll call gene trees, to each other and to a genome-level tree, trying to look for patterns within these trees and reconcile these two different scales with which we might want to estimate evolutionary histories. And I want to quickly give an analogy. So let's say I have one genome tree and I have 20 gene trees. My goal is to come up with a 2-dimensional visualization where I can have points that represent each of my trees, and that give me information about their phylogenies as a whole, and I can look for patterns. So here, based on this plot, I could say that gene tree 1 and 20 are similar to each other, and that they're pretty different from my genome tree. So hopefully this idea is pretty familiar to a lot of you. This is a similar idea to what we actually just saw in the previous talk. This looks very much like a principal coordinates analysis plot that we often see for when we look at microbial compositions. So in this case, we might have a variety of samples. Here, imagine we have 20 samples, and we have counts of how often we see a variety of taxa throughout these samples. So often, instead of looking at how these samples are similar or different based on just taxon 1 or taxon 2, we perform principal coordinates analysis to get a visualization in 2 dimensions that incorporates the information from all of these taxa. Again, we can compare samples by say sample 20 and 2 are similar, they're more different from sample 3. So the work that I'm doing here is actually quite similar. The differences are that instead of wanting to plot our samples as our points, we want to plot our phylogenies, our gene trees, and our genome tree. And instead of using a vector of counts as our data, we're actually going to use all of the information encoded within our phylogenies, our branching structure, and our branch lengths. And the way that we do this is we start by estimating the gene tree for every gene using those gene level multiple sequence alignments. And we also estimate a genome level summary tree. We use concatenation for this, but you can use any genome level tree that you'd like to, as input to our tool. We then map our estimated trees from VHV tree space to a Euclidean space. What this means is that these trees, which recall they have their structure, their branching structure, and their branch lengths, can be perfectly embedded into this complex non-Euclidean space that we call tree space, but it's really hard to do math in this space. So we come up with an approximation for each tree into a high-dimensional Euclidean space. This is the normal space that we like to do math with, and then we can reduce the dimension of that space using principal components analysis, quite similar to principal coordinates analysis, to visualize our set of trees in a low dimension and come up with a simple 2-dimensional plot. So an example of this is an analysis here that I have of 73 Prevotella genomes from the GTDB, and we are analyzing 63 genes that are each contained in all genomes. So here, this plot is similar to the toy example I showed earlier, where each point in my plot represents a tree. The ones in black are gene trees, and the red one is my genome level summary tree. And their position on this plot is determined by information coming from the phylogeny. And I think the most interesting thing about this plot here are the outliers. So we can see that in our first coordinate, we have 2 genes that show up as clear outliers. And if we actually go ahead and look at the phylogenies for these genes, we see one very long branch, which leads to the same tip 51, which you can't really see at this scale. So this means that we might want to investigate these genes further. It's possible there's some biological signal just for these genes. That means there's a lot of genetic difference for this one genome compared to all the rest. But it's also possible that this is an artifact of the data. I think more interestingly is the outlier in the second principal component. This is the gene BAC-A. And again, we see one very long branch in our tree representing a large amount of evolutionary distance, but this is between 2 different clades of tips in our tree. So it would be interesting here to go further into this analysis and ask, are there any differences between these 2 sets of tips based on perhaps the gene BAC-A, which is a gene that confers resistance to the antibiotic bacitracin. So my point in showing you this analysis is an example of how you might use this tool. So here we were able to identify gene level phylogeny outliers. Essentially, we were able to take what was a ton of information. I think we had 64 different trees on 73 tips each, and distill it down and look for what are interesting signals. And then these signals could be a jumping off point for a future analysis. So this could be the quick plot you make at the beginning to figure out what can be interesting at the gene level for evolution in this set of genomes. You might also use our tool to contrast multiple sets of genes. So here I have another analysis where we look at a set of Streptococcus genes. And here we compare across a covariate. We look at a group of ribosomal genes and compare that to a group of other genes with more diverse functions. So here we can see, just like your typical principal coordinate analysis plot, we can color our points by some sort of covariate we're interested in and see if there are any patterns in our data. And finally, we have an example where we use this tool to highlight anomalies in pre-processing of the data. So here I have a different Prevotella analysis. And we actually ran this analysis twice. The first time we estimated all of our trees, both our gene level trees and our genome tree. We found that, interestingly, our genome tree was quite different from all of our gene trees, which suggests that there's some kind of signal in our genome tree we weren't seeing in any of the gene trees. So to try and figure out why this is happening, we started by just running all of the analyses again. And the second time, we saw that the genome tree was actually quite similar to the gene trees. And after seeing this, I went back to the first run of the estimation algorithm and saw that it had never converged. So really, this first genome tree was a pretty bad estimate of our genome level phylogeny. So here, there are other ways we could have figured out that something strange was happening, but if we had only looked at our one genome tree from the original analysis, we might not have known that it was a bad estimate. So here, by actually making this plot and comparing a genome tree to all the gene trees, we put it in context, and something that could either be an interesting biological signal or could be an anomaly in preprocessing really stuck out to us. And conclude by talking about some limitations. The main limitation is that we can only consider genes that are in all genomes of interest. This is a problem biologically because not all genomes contain all the genes we care about, especially if we have a more diverse set of organisms that we want to compare. And additionally, when we sequence, we might not actually get data or usable data for all the genes that we care about for each genome, even if those genes exist. So this means practically for this tool, you have to subsample your data. So you have to choose which genes and genomes you want to be included in your study, and then build up a larger set of genes and genomes based on that. And we have advice on how to do this in a tutorial for our R package. So to learn more, you can see our paper on this project, which is recently published in the Biostatistics Journal. And you can also refer to our R package called Groves, which is available on GitHub. I'll conclude by thanking my advisor, Amy Willis, as well as our collaborator, Mike Lee. And I also have my email here, and I'd like to invite you to reach out if you have any questions about this work or ideas, either about how you might use this, if you have any ideas about different analyses you could do with this tool that we haven't thought about. I'd love to hear them, or ideas on how to improve this.